Assalamualaikum. For this week, I'm going to teach you guys how to design your research properly. Why do you need to do the research plan? Why do you need to plan your research properly? And how to design your experiment. Designing your experiment, your research properly based on your question and hypothesis will give insight on what type of statistical or biometric analysis that required in your study. Follows that, I will introduce you guys type of experiment that required based on your type of studies. Of course, last week we have discussed on different type of experiment or different type of biological studies, and to this week we're going to focus on assays that commonly used in that particular study. So I'm going to introduce you guys to how to choose type of assay according to your studies, and then what are the number of replicates, or the repeats, number of samples, the important, all these fundamental basics in your experimental design. How choosing the number of replicates, number of sample, and having control in experiment will give you an insight into your data analysis, and also give you guys precautionary steps before committing some sort of what we call an errors in your decision. Okay, of course, when we do the analysis, we observe a biological phenomenon. We want to report an actual biological phenomenon. We don't want to misinterpret our data. Later on, we find that our data turn out to be our analysis, our decision, our conclusion that we made based on our data is not correct, do not represent the biological phenomenon. So how to avoid that? Okay, for this week, I'm going to introduce you guys there are two types of errors in the biological studies, which is the type 1 error and the type 2 error, and how to avoid that. Okay, so when you starting your research, okay, maybe some of you already start doing your research already. Some of you have to stop. Some of you doing it computationally. There are things that we have to be aware of. We don't want to misinterpret our finding. The whole reason is we want to report the actual biological phenomenon occurrence in the nature. We don't want that later on we found that our data does not represent the biological phenomenon or the natural phenomenon turn out to be due to some technical errors or due to the mistake during the analysis. There are a few steps that you have to do in order to avoid misinterpreting your data. The first thing first is you guys need to include the negative and the positive control in your experimental data. Okay, the negative control, as I have to stress, negative control is always a must in every type of experiment. For positive control, if you are doing something that are standard, positive control is a must. Okay, in any standard procedure or any what we call this a very stringent lab practice, having both negative and positive is a must every time when you do the analysis. And also make sure when you do the analysis, when you run your sample, make sure that you prepare, you have enough replicates and or number of sample for that's much bigger power when you do the statistical analysis. Okay, and then after that, you need to repeat the experiment several times. Okay, minimally, ideally, you have to repeat 3 times. Okay, the reason is you want to make sure that your data are very basically that are consistent, to make sure to confirm that it is the natural biological phenomenon. You don't want, if you have to replicate on the first experiment, those data you get probably could be caused by chances, right? And if you repeat again and you still find it, you get the very similar result and you repeat again, you got something similar. By having those, you could rule out that your data is not by chances. We will explain later on in the following weeks. So there are 2 types of control. There are positive control and the negative control. Usually, by definition, positive control is a condition in experimental data or assay, for example, like PCR. When you do, it will definitely show a positive result. Okay, it has to be something that already been known before. Okay, you have to use as a positive control. And then you have your treatment. And then, of course, the last bit, you have your negative control. Negative control is a condition where you have your sample, but that particular sample, you don't do any form of treatment. That is your negative control. For that, by having the positive and the negative control, you know the performance of your growth, whether it is whether it is better than the negative without treatment, or basically it does not meet the negative control. Allows you to make sure that your performance, the treatment, does works against the negative control, does work against those who are untreated, while the positive control will give an insight that you are not committing the false positive finding. Having these two controls is very important. I could stress more, it is very important in any form of laboratory experiment or greenhouse experiment, or samples that you took from the field and then you do the further analysis in the lab. Okay, these are required, is a must. Having a control is a must. Okay, so when you do your finding, the reason here is you have this positive and negative control. You want to make sure that you are not committing the false positive or the false negative finding. Okay, you don't have a false positive result. If we look through here, you have, for example, you have your control here. The negative, you have the negative result. Okay, this is your control negative. This is your positive control, and this is the treatment of your interest. If we're looking at here, I give you an example. We have, for example, in the plant treatment, you have a set of plants. Okay, this is treated, and this is untreated. Okay, and you have 3 plants. 1, 2, 3. You give treatment on this one, on this plant, 3 treatment. Okay, and of course, you treat, for example, you treat it with the hormone A. All right, and of course, this untreated is considered as your negative control, while you should have another treatment. For example, treated, but this one we treated with the hormone C, but C has to be known to give a better performance or better yield or better growth on the plant. Okay, and these are your positive control. Okay, so by having positive and the negative control in your experimental setup, you will find that if you get your data, when you see the performance equivalent to your treated of interest or to the positive, and at the same time your positive control also showing the better growth, same cases, and you're treated, your data probably a false positive data. Okay, in another situation, if you are turn out to be your negative control have a positive result, okay, you have to see something growth, just a growth here. Somehow your positive control here don't show any performance or much worse compared to the negative. However, your positive, your treatment here does show better performance than the positive, than the negative. And the positive, what does it mean? You are still committing a false positive finding. Okay, so how to make sure that you are actually, your finding here, the treated does actually work, is when you see a better growth performance of your treatment compared to the compared to your negative control. At the same time, it also, you would find that it does grow about the same rate or slightly lower, depends on the treatment. If they're better than the positive control, then you know that your treatment is actually work. Okay. Another situation is when you have your treatment, in this instance, you don't see any performance between your negative control and your treatment, doesn't show any differences in terms of growth, while your positive control have a better performance, it does growth better, produce more yields. This indicate that your treatment for the dose A and for the hormone A does not work. Okay, it depends. Sometimes it could be good news, sometimes it could be a bad news. But it is your finding, that is your data. All right. Another situation, you have a better growth. You find that your treatment with the A treated plant and your negative control, your positive control, do not show any better performance or have a performance growth lower or slower compared to the negative control, does indicate probably your treatment failed or you're probably committing the false negative finding. By having this negative and a positive control, it definitely gives you a finding indicate that your treatment is actually a true finding based on the biological phenomenon. It is not due to the false positive. Okay, another situation here, this is much more worse. If you have negative control, somehow your negative and your positive control do not show any performance, while your A treatment have a better growth, you're probably committing either false positive or negative finding. Do not differentiate between your actual finding or between compared to your false positive or your false, whether it could be false positive or could be a false negative. Okay, but if you don't see anything at all in all from positive control, negative control, even your performance, you're actually committing a false negative. Okay, and in this instance, you need to look back into the technical error, how whether you don't use the proper plant or you don't do the timing correctly, or you use the wrong chemical, or the fertilizer is already expired, and many more. There are many reasons we have to look back why the treatment doesn't work, which is supposed to work. That's why having the positive and the negative control is important. I'm going to introduce you guys replicates versus sample size. Two types of replicates. We have the biological replicates and we have the technical replicates. Well, how is it important? Biological replicate is a parallel measurement of biologically distinct sample that capture random biological variation, which may itself be a subject of study or a noise source. While the technical replicate, by definition, is a repeated measurement of the same sample that represent independent measures of random noise associated with protocol or equipment. And the reason why you need to do the technical replicate, it will be useful because it will be an indicator for technical error and also indicate the consistency of your assay. All right. Usually, the symbol that we use for the biological replicate or any form of replicate, we use the N symbol. If you have 3 replicates, we use N equal to 3. So, what's the difference between the biological replicate and the technical replicate? So, if we have in distribution, okay, in the population, we have, for example, we have a mouse strain. For example, in this instance, we have the Bobsy mouse. So what happened here is we have mouse here, a whole set of mouse. We only take 3 mouse here, and then you extract bone marrows from 3 mouse. Okay, from these bone marrows, of course, the reason I have 2 here, right and left, right and left, right and left. Okay, so these are first replicate, these are your second replicate, and these are the replicate. Okay, so anything that coming from biological organism, okay, is considered as a biological replicate. So when is the technical replicate? For example, you have this sample 1 here. Okay, you take the cell from the bone marrow, grow them in a plate. 1, 2. So what happened here is coming from here, and then you do another. Of course, each one of these 1, 2, 3, 4 coming from here. This indicate a technical replicate. 1, 2, 3, 4, because coming from the same biological, and you seed it into 4 wells, that indicate these are the technical replicate. Because what happened here is you measured based on the 1 mice here. So these are your biological replicate. For the technical replicate, you have this and you further split into, for example, like further into 3. Right? And these are the technical replicate. Okay, you have to make sure it has to be coming for the biological organism itself. But technical replicate from one biological replicate, for example, from one biological replicate, you further do another. You seed it into different wells before you do analysis. Those are considered as a technical replicate. So when should you use biological replicate and when you should use a technical replicate? If you are doing experimental in the lab or in the greenhouse, it is important that you should have a biological replicate. Okay, and ideally for in any scientific publication, not just in your thesis, for publication in the journal, you should have at least 3 replicates and you should always do 3 times repeats. So basically 3 times 3 indicate that you have a n of 9, which is indicate a 9 replicate. Okay, so you will repeat 3 times. You have to replicate, repeat 1, second time, the third time. So in total, you have number of replicate equal to 9. Okay, it is a must for in vivo and then a must for in vitro ex vivo experimentation. All right. So by having more replicates, indicate will give you more statistical power, and by having more statistical power allows you to reject your null hypothesis and also avoid from committing false positive finding. All right. And while the technical replicate is to assess, the whole reason is you want to assess your assay efficiencies. So if you usually using PCR or any form of, for example, if you count the number of cells 3 times, those are the technical replicate. All right. And these are to measure your efficiencies and the consistencies in the finding. Okay, so what are the signs when you need to do the technical replicate? When you find there are discrepancies in your data. Somehow you have your family, you usually do this experiment, and somehow you do it many times. Somehow when you see there's something wrong in your data, that is definitely you need to investigate what's going on. Okay, if you're rich, you can just toss the equipment, toss the reagent and use a new one. Okay. Sometimes we don't have, we have very limited resources. Sometimes we need to find out what are the costs. Sometimes we have to use a technical replicate approach to find what are the sources of your errors. Okay, and this is fairly important, especially when you receive new batch of kits of chemicals. Okay, sometimes when you are being cheap, you have very limited resources, you have a budget, constrained budget, and you definitely need to test your kit. So for this, you need to do the technical replicating application. You need to use the technical replicates in this instance. All right. Okay. So what are the difference in comparison? So for the sample size, usually when you do field sampling, you go to, for example, you go to the forest reserve, hutan simpan, and then you collect sample. Okay, over there. Or you basically do some sort of observation, you record the number of species over there, or you do the image capture. Okay, or you actually capture the insects or the birds or any form of plants or whatever it is, and those are considered as a sample size. Okay, those are considered as samples. All right, so anything that goes from the sample, from the sampling, the actual collecting the collecting the sample from the field or from the outside, from the actual distributed population, those are considered as your samples. Okay. So the sample size are fairly important when you are doing a field sampling study, when you do field sampling works in a clinical study or in the population survey, right? And the whole important bit is you want to make sure that the number of samples that you have should represent the observed population, and also it should be enough to reject the null hypothesis while not committing the false positive. Okay. And this can be done through power calculation. We're going to discuss this more next week. How to calculate power calculation. Right? Okay. So having the enough number of replicates and also enough number of samples, if the whole reason is you want to avoid committing the false negative or false positive conclusions. Okay. So the reason is having the reason we call false positive and false negative. It lead to the misinterpretation of the actual biological phenomenon. It does not, your finding somehow either contradict or do not agree with the actual biological finding. Okay, and this is detrimental if someone else finding that are against contradicting from your findings. Okay. Another name, if you are in the statistical analysis, be the false positive, the type 1, type 2 error, also give you a wrong inference of your null hypothesis. Okay, there are many reasons that can cause type 1 and type 2 errors. Probably usually it's due to your wrong experimental design or setup, or usually you don't have enough sample replicates. Okay, so what is type 1 and the type 2 errors? In the hypothesis testing, type 1 error is considered as alpha, and another name is false positive. Type 2 errors is usually we call it a beta, and also known as a false negative. All right. So if we look through here, we have your null hypothesis, which is in reality, or your reality is, and you have your true, which is your true hypothesis, true reality, and this is false reality. Okay, these are the false. Okay, so we have on our H null. So what happened here is we find that our findings somehow accept our H null, and the reality also agree with the H null, with your finding. So these are considered as a correct inference. Okay, and this is the actual true negative. Somehow what happened here is, if your finding is somehow turn out to be disagree with the reality, or the reality does not show the same, you are basically have committing a false negative, or another name is type 2 error or beta. Okay, while if you reject the null hypothesis but turn out to be in reality is to be true, you have committing the false positive error. Okay, another name is a type 1 error in the hypothesis testing. All right. Usually what we want here is your hypothesis null. If you rejecting and you also find that in reality it turned out to be also false. So you have a correct inference, and these are the true positive. So when you're rejecting the hypothesis null, of course you're accepting the alternative hypothesis. All right. So how to remember this false positive or false negative is, if you are accusing someone innocent to be guilty, if you are a judge, you are accusing someone innocent not to be guilty, you have committing a false positive. Okay. While for the false negative, you're proclaiming someone guilty, well, they are innocent. Okay, you're proclaiming someone guilty, being innocent. They are turned out to be, they are guilty, but you are freeing themselves. You said that they are, he is innocent. So basically have committing the false positive. Okay, or type 2 error. Thank you.